this DNA microarrays in terms of the concept of what these microarrays are and how they are applied. Okay, and although at first glance it might appear that it may not be of interest to us, it is of interest to us, as you will see a little later. Okay, so we will begin our story with the discussion of what we call as a central dogma. Okay, which is the way information flows in biological systems. And we said that most of the information in biological system is stored in form of DNA, and this DNA serves two functions. One is a replicative function, where it faithfully copies itself in preparation for passage to the progeny, and the other, which is called as life functions, where the information in the DNA is read initially by transcription to get an RNA, and from that RNA it is translated to give you a protein. Okay? And this is a very classic flow you have seen probably many times now. Okay, the double-stranded DNA, the single-stranded RNA to the final protein. But this is only kind of part of the story in a sense, because if you think very carefully, when DNA is read to give you RNA and then to give you the proteins which are actually produced can be of different types: structural proteins, then receptors, then enzymes, then other proteins. Okay, many of them whose function is probably still not known, and they tend to come together to form what are called as the different biomolecules which are present in the cell. Okay, so when you learn biochemistry, you also learn that synthesis of lipids is carried out by enzymes from small starting materials. Similarly, carbohydrates are also fixed by using sunlight and enzymes. So all these biomolecules are all made by the function of many of the enzymes, which again is produced from your DNA information. And these biomolecules come together to make up what we call as a cell. Okay? So in a cell, you'll have all these lipids, carbohydrates, proteins, and nucleic acids. Then cells come together to become tissues. Tissues come together to give you the organism. And again, this organism carries out the two main functions, again, via DNA and its information, that is replication and life functions. Okay. Now this is very common pathway that occurs all through the biosystem. Okay, and every organism does this in one way or the other. Okay, and a field of study which looks at these aspects in a more global fashion is what is called as the field of genomics and proteomics. Okay? So what is genomics? When you look at everything that relates to nucleic acids, okay, DNA and RNA, in terms of the structure of DNA, mutations, okay, like deletions, insertions, when the DNA gets expressed, where it gets expressed, where is RNA formed, what is the extent to which RNA is produced. All of these aspects and their studies come under the umbrella of what is called as genomics. But once you get the protein, to look at the proteins, how it is produced, how much it is produced, which proteins are produced in which cells, what is the function of all of these proteins. That comes under the ambit of what is called as proteomics. Okay? Now, with respect to only genomics, okay, basically what genomics does is that it integrates the nature of the genome, okay, and also the genes which are present on the genome and their functional expression and the consequences of the expression of genes. Okay, so like in a sense, if you were to look at genomics, some of the questions it is trying to ask or it's trying to answer is that what is the genome sequence of a particular species and which are the polymorphisms that are observed in the different members of the same species, okay, which makes each member of the species different from the other. Okay, where are or what are the gene or gene-like sequences in the genome, and how many such sequences are present? Okay, that means a given genome of an organism, how many genes does it have, and then in which specific tissues are which genes expressed. Okay, that is, if you look at it from a cell perspective, different cells will express different genes So what is the complement of the mRNA, which is called as transcriptome of a particular cell? Is this expression altered during development? Is expression to stimulation, pathogens or drugs, or is it changed by exposure to pollutants, etc.? How does expression of one gene affect other genes? Okay, can we relate the gene expression profile of a particular cell or a tissue to a disease state? Okay, and then what are the products of the expressed gene? All of these questions are typically questions which are asked when you are talking about the genomics aspect of understanding a biosystem. So, what is important is that you should realize that the function of a given cell and therefore the related tissue and the organ which the tissue ends up making, and the organ system and the organism are all governed by the set of genes that are present at a given time. Okay, so as we are sitting here listening to this, our status is actually governed in a sense by which cell is making which genes, and therefore overall how this cell's functioning is affecting our own system and the organism. And we also have to realize that the expression of genes or the activity of genes is sensitive to both what is called internal and external stimuli. Internal stimuli like, say, hormone signals, autocrine, paracrine, endocrine, and external stimuli like, say, infections, pathogens, exposure to chemicals, and so on and so forth. And thus, this change in gene activity as a result of all of these internal external stimuli can result in altered transcription and then later on altered translation or even modifications. Okay, what is called as post-transcriptional and post-translational modification status. Okay, so given that analysis and cell behavior under what we call as normal or what is called as disease states. And not only that, if we can measure or analyze gene activity of a cell, maybe they'll give us a handle of how, because of that, a tissue behaves in a certain way, or an organ behaves in a certain way, or how an organism behaves because of changes in gene activity. Okay? So, so this area of genomics, where we try to measure or analyze the genetic activity, is a very happening area of work. Okay, and there are several ways people have attempted to study changes in what is called a gene expression. Okay, and here are several methods for looking at it. For example, what we call as northern blotting, or what is called as RNase S1 nuclease protection, RT-PCR, or READS, as it is called, restriction enzyme analysis of differentially expressed sequences. And in addition to that is this way of analyzing gene expression, that is by the use of what are called as DNA microarrays, also called as DNA chips, or gene chips, or what is called as genome chips, etc. It goes by different names. So today, what we are going to do is look at this aspect of how this technology is used to address this interest of ours, that is measurement of how genes are expressed and how this expression is changing under different circumstances. So, what are array, or what is a DNA array? Array by definition is a systematic arrangement of samples. Okay, so in that regard, a DNA array would simply be a systematic arrangement of samples whose nature is, uh, whose composition is DNA, or whose composition is what are called as oligonucleotides. So when you have a systematic arrangement of different DNA pieces or different oligonucleotides in a very systematic fashion, we call it as a DNA array. Okay, so typically what you'll find in DNA arrays are oligonucleotides which are anywhere from 18 to 100 nucleotide long, or what we call as 18 to 100 MERS. Generally, you'll find it somewhere in the range of 25 to 30. And these are arranged on usually a glass slide or a nylon blotting membrane. Okay, so imagine for a second that you have a glass slide and you have got at different places on the glass side, uh, arrangement of different types of DNA. You would call that as a DNA array on a glass slide. Now, when it is called a macroarray versus a microarray, is just depending on the size of the spot. So, if you have a spot whose diameter is 300 micrometer, or I should say radius is 300 micrometer or more, we will call it a macro array. And if the spot size radius is less than 200 microns, we call it as a micro. Okay. So pictorially, it will be something like this. So here are different spots you see on a little slide, and each spot here has different DNA sequences. So if I were to go to this area here and look at all the DNA which is present in that area, they'll all have the same sequence. Like for example, AAA, TT, GCA, TA, GCA. Okay. And usually these DNA microarrays that we will talk about are composed of these slides, or what you call as membranes, where the number of spots are in the range of thousands. Okay, and each of these spots will be of a different type of DNA in terms of the sequence. Okay, so that is kind of conceptually what is a DNA microarray. And if I were to, take you further, go near the spot, you will find that in a certain region, all the DNA will be of the same sequence, and it is somehow attached to the glass plate or the nylon membrane. Okay, most of the time we try to attach it covalent so that it doesn't run away. Okay, and so in each location, you'll find there are millions of DNA strands. Okay, and like that there are thousands of locations. Okay, so what I mean is, in this region here there will be millions of molecules which are having the same sequence, and like that there are thousands of different spots. Okay, so that is what constitutes what is called as a micro array. Okay. Now, what is the principle which is involved in the use of what is called as DNA arrays? The principle is very simple. Okay, it is based on what you have learned as Watson and Crick base pairing, which says that if there are two sequences of nucleic acids, either both DNA or both RNA or a DNA RNA hybrid, if they are complementary to each other in terms of their bases, that is AT or AU and GC, okay, they will hybridize under what are called as permissive conditions to yield a double stranded structure. And we know of this, okay, that if I take DNA which is all A, and to that I bring a DNA which is all T, A and T, because of their base pairing tendencies, will come together and form two hydrogen bonds, and they'll form a double stranded structure. Okay. Now, with respect to the DNA microarray, the DNA which is present on the glass slide, okay, which is this DNA here, okay, which I put again on the spot. Okay, these DNA molecules are all referred to as probes. Okay? And the reason they are called probes is because they are actually going to locate a complementary DNA or RNA in a sample, which is the target for this probe. Okay, for example, so if I have at a certain location here all A, T, T, G, G, and say this is 5' to 3', then this spot will have a tendency to hybridize with all the DNA in the sample which has got a complementary sequence, that is T, 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 A, A, G, C, T, A, T, C, G, and T. Okay, because of this complementarity. Okay, so that is a target for the probe which is present on the slide. Okay, now if in your sample you are looking for DNA to be targeted, then this interaction between DNA and DNA is referred to as southern blotting. Okay, and if in your target you have RNA which is going to hybridize to your probe, which is a DNA, then it is called as northern blotting. Okay, now the issue is, all we have to do is characterize how much of this double-stranded entities are being formed. Okay, uh, and that will tell you how many targets are present in your sample. Okay, so the issue is to allow hybridization to occur and somehow characterize that hybridization so that you can get information on both the nature and extent of the target. Why nature of the target? Because if I find this is occurring at a specific spot, okay, here, because I know which DNA is here, then I know that the sample has is something which is exactly complementary to that. So I get an idea of what is the nature of the target. And second, how many such events are occurring here will allow me to even quantify the number of targets which are present. Okay, and that is how we are able to do what is called as gene expression analysis. So what we'll typically do is look for mRNA in your sample, which will tell you how much the gene is being expressed, or you take the mRNA and convert it into DNA, what is called as copy DNA, and then use that DNA as a surrogate for the mRNA. Okay, so we'll look at how that is done in a systematic fashion. Okay, so this is the idea. So if at a particular location I've got a certain sequence and that is called as a probe sequence, then it is also always going to bind to something which is complementary, which is present in the target, and that is called as a target sequence. Okay, and all we have to do is somehow analyze for this binding event when it occurs in terms of how much is occurring and where it is occurring, so that you know what is the nature and the quantity of that target which is present in the sample. Okay. Easier said than done, but typically you need to do a few steps so that you can use this technology correctly. So typically you'll find that when DNA arrays are used, you need to do about 5 steps, okay, so that you can do this gene expression analysis. The first thing is you have to prepare the DNA array, okay. Uh, Second is you have to do what is called as test sample preparation. So you have to do what is called as sample preparation, as we say. And then once you have the two things ready, the array and the samples ready, then you actually do the hybridization. And after you allow the hybridization to occur, you detect and find out how much it has occurred and where all it has occurred, so that you get idea of what is the nature and the concentration of the target, and then you quantitate that. And once you do that, then you look at the data and then you do the data analysis and the interpretation of what you are seeing. Okay. So we'll look at this in a very systematic but quick manner. Okay. How do we prepare a DNA? Okay, there are two basic approaches to making what are called as DNA chips or DNA microarrays. In the first three methods, which are put like with the underline, what you do is very specifically using what is called as solid phase DNA technology, which is now very well known and very well accepted. You synthesize all the different types of probes you want to make, okay, separately in a in a container or a beaker or in a test tube, okay. So essentially what you have is, based on your synthetic skills, a whole number of pure samples of different sequences. And once you have that, you very systematically take a small quantity of each of these different types of DNA, and using either what is called as micropipettes or what is called as inkjet technology or what is called as drop test technology, very systematically pipette them out or print them out on what is called as a, a glass slide or a plastic slide or a membrane. Okay, and that way you know, okay, you have got different spots containing different DNA, and you know, depending on where you pipetted what at what location which sequence is present. Okay, so that is kind of shown here. So you make your DNA and then you load it, okay, and you do what is called as contact printing, like inject printing as it is called, or you take a little needle and you put a drop wherever you wish, okay. And the way you do this printing or this dropping, if you do that systematically, you know at all the different locations of your glass slide which sequence ka samples are present, okay. So that is one approach, that is make DNA separately and then take this prepared DNA and then spot it on the glass slide or glass plate. Okay. The other approach, which is called as the Affymetrix approach or the nanogen or nimble-gen approach. In this, what you do is you actually do the synthesis on the glass plate itself. So you don't really make the DNA separately, but what you do is on the glass plate, in a very systematic manner, you synthesize different sequences at different locations. Okay, and again, the technology for synthesis that is used is same as what you do for synthesis in a test tube, except it is now done directly on the plate. Okay, so what we mean now is that the chip is actually fabricated in situ. That is, the sequence is built using what is called as photolithographic techniques. Photolithography is light induced what is called as etching. Okay, now I'll show you how this is done. Okay, it's very elegant. Okay, so let us say I have a plate here, and on the plate I've got these little linkers which are already loaded. Okay, and this is a pre-synthesized plate where these linkers are present on the entire surface of the glass slide or silica plate or the membrane. Now what I do is that I bring a little sheet on top of this plate, and on this sheet I got little places where there are holes. Okay, and so when I put this sheet exactly over this glass slide and I shine light, because there are holes at certain places, the light will go through that region and hit the actual surface of the plate. Now, the wavelength of light is chosen in such a fashion that it now does a reaction on the surface of this plate in that very location where it hits the plate. Okay? And what it essentially does is makes that region of the plate reactive. Okay, and that is shown here now, that what is present as a O protection group is now removed and you get a OH. Okay, now once you have a OH, what you have essentially done is functionalize that region of the plate. Okay, while all the other regions still have this little protecting group because no light has passed through them. Now once you have activated this, what you can do is, depending on what sequence you want to build, bring the correct base. Okay, let us say in this case I bought T. Okay, so after I bring the T, I know that this T is only going to react here because that is the only surface which is open. Okay, now see what I do. After I do this addition of T onto the surface, I again put a leather sheet, but here now the hole is at a different location. And now when I shine light, it activates this region here. So I get OH, and then when I add C, I'll get a C there. Okay. So you see how I do that. I make these little sheets with holes at different places, depending on what sequence I want to build up where, and very systematically by doing this light induced protection and deprotection, I actually make up the sequence. Okay. So just to give an idea, okay, let us say after the C was done, in the third step, what I would have done is I want to add G in this location, right? So this location still has a O. But see what I do. When I do this step for adding G here, what I also do is make a hole here. So when I make a hole here, what will happen? This T group here will be activated in addition to this. So I'll get TOH and a OH. So when I get TOH and a OH, when I add G, the G will be added here and another G will be added at this location. Okay. So you see how I will adjust my holes and all. So this is what it is shown here. So I prepare this different what are called as masks, where there are holes at different places, and when I shine light, it will go right through this region only. Okay. So basically what is done is that you know what is the length and breadth of your glass slide or your membrane or your plastic support. You also know at what location you want which sequences. So you can actually tell the computer, okay, and it will write a little, you can write an algorithm by which the computer will tell you how to build up the sequence using different of these what are called as masks. So it will tell you, first use a mask of this type. After you are done, then the second mask will have holes at different places. And the third mask will have holes at different places. And it will allow you to then very quickly build up the sequence in a very systematic fashion. Okay. So, so this was kind of a breakthrough technology which was actually invented by Affymetrix. That's why this photolithography technique is also sometimes referred to as affymetric technology. Okay, but the big drawback of affymetric technology is that tomorrow, if I want to make a glass slide with sequences which are different than what I just did, then I have to make a whole new set of these masks, because now the way the sequence will be built up will be different because the sequences are different. Okay, so the affymetrics technology worked great if you are going to make the same type of chips again and again. But the minute you want to make different types of chips, then the cost goes up, because now you have to tell the computer again all the new sequence that you want, and it will actually prepare an algorithm and tell you what are the masks to use. Okay, so somebody said that, rather than doing this mask approach, let us do a slightly different approach, and this is very interesting approach, which is done by what is called as NimbleGen, what is called as digital light processor. Okay, so here what they do is use the same technology as it was used by affymetrics, but rather than using a mask with a hole through which the light is shown, what they do is something very similar to what you see on the playgrounds. If you remember, when you see day-night matches, no, and they take the camera far and they show you the ground, you notice something in a day-night match. What do you typically notice when they take the camera away from the ground and give you the bird's eye view? And let's say it's 8 o'clock at night. Immediately you'll spot something. The ground is all lit up, and the ground is lit up because of these four pillars which have got a whole bunch of bulbs whose light is now thrown onto the ground. Okay, but you all know that if you were to closely look at these four pillars on which the lights are mounted, or what are called as the stands on which the lights are mounted, there are numerous number of lights on each of these towers. Okay, and the idea of doing that is because tomorrow, even if one light kind of fuses out, the remaining lights will still be there. Okay, the last thing I want to have is a big bulb shining light onto the ground, and if something happens to that bulb, there will be total darkness on that tower. So rather than doing that, they use a whole number of bulbs. Okay, so now imagine for a moment that you have control over the orientation of the bulbs which are present on the four towers. And if you had that control, what you could potentially do is move all of those lights in such a fashion that all the people only in the east stand will be shown on the light, and everywhere else there will be darkness. Okay, it's kind of what they do in a lot of these stage programs, where they have this lighting and they switch the lights on and off, and they can focus the light exactly where they want on the stage, or on a very specific member, or on a person who is in the audience, whichever they want to do. Okay, so what NimbleGen did is that they said, rather than having masks and shining the light in such a way that it goes through the hole in the mask, what I'll do is I'll keep the light on these towers which will be present on this side here, and I'll adjust the mirrors on that tower such that when the light is shown on the mirror, it will be reflected and go exactly where I want to. Okay, so you see what they did? Rather than using masks, what they did is they made what is called as a tower with a lot of mirrors on it, and these mirrors were individually movable, and when the light was shown on the mirror, the angle of the mirror was adjusted in such a fashion that you could get light shining wherever you wanted. And the big advantage of this is, tomorrow if my sequence is changed and I want to make another sequence, all I have to tell the computer is how should I move the mirrors, so that I can accomplish that task rather than making new masks. Okay, so very elegant technology of what is this company called NimbleGen. The idea was to, rather than make DNA separately, just make it on the chip wherever I want, whatever sequence I want. Okay, so after we are done, we'll typically find that at any one given location, we'll have multiple strands of DNA all having the same sequence. And generally, the probe sequences that are used are about 20 MERS long. Okay, and what you'll typically find is, in one location, there'll be about anywhere from 2 to 2.5 million of these exact same sequences in a given location. Okay. Now, why are we choosing a 20 more? Okay, and okay, before you go that, in reality, no, if you were to look at the DNA microarray and do what is called the electro micrograph, you actually find strands like this. Okay, so they're not like standing up like this, waiting for the other DNA to come. They just flop out. Okay, there's nothing. It's a backbone नहीं है उनको. खाली sugar phosphate backbone बोलते हैं अपन, but they are not like you and me, able to stand straight. They'll just flop on their backs, and actually in an electron micrograph they look like this. But in a one location, all of these strands will have the same exact sequence. That is the idea. Okay. Now, as I said, typically these little slides that they use are very small. Look at this, 1.28 centimeter by 1.28 centimeter. So just like 1.5 by 1.5, and see how many different probes are there. Okay, 2,500 different probes. Okay, which means that every location here is about 24 micrometer in diameter. We have multiple spots. Collectively speaking, in this small square of 1.28 by 1.28, you can have 2,500 different fields. At each location, there's a different DNA. Okay, so they are highly miniaturized, but very information rich. Okay, now why do we use a 20 more? The reason we use a 20 more is simply this. If you look at the probability, okay, let us say I have a 20 mer of a given sequence. Okay, so the chances of that sequence occurring at random, okay, let us say that sequence is ACGT, again ACGT, again ACGT 5 times. Okay, that give me that 20 more. The chances of that sequence occurring in random in a piece of DNA is simply this. Because if I look at the random uh, occurrence of a 20 nucleotide base sequence, then the probability at that one location, it could be A, is 1/4. The probability at that second position, it's a C, is also 1/4. The probability at the third position is a G, is also 1/4. So the probability that is ACGT, again ACGT, is 1/4 raised to 20 times, which is nothing but half raised to 40 or half raised to 10 raised to 4, or 1 divided by 1024. 1 raised to 10 is 1, 2 raised to 10 is 1024, again raised to 4, which actually works out about 10 raised to minus 3 raised to 4, or 10 raised to minus 12. Okay, so the chances of this 20 base sequence occurring at in a human genome, which is about 3 million base pairs, is 0.003, or what do you say, 0.03% chance that will occur at random. Okay, and that's why we chose this 20 mers, because a 20 mer sequence occurring by chance anywhere has got such a low probability that if you choose the 20 mer correctly, you'll be able to exactly spot the gene of interest to you, because that sequence has got very little chance of occurring in any other gene. So that's where the specificity comes. So if you design the probes correctly, the sequences correctly, each probe will allow you to attract a very specific mRNA or a very specific target DNA, and therefore we will know exactly which gene you are analyzing in terms of increasing or decreasing the expression. Okay, so that's the reason we choose a 20 more. Okay, so now the second part is how we prepare the sample. Okay, where do you get the tissue sample? Tissue samples can be obtained from various places. I can get cell lines as a source of my RNA, and I'm looking at transcription in the cell lines. I can get tissues from a animal. Okay, Sandhya would remember her M-Farm project. She was doing induction. Okay, so imagine that you gave a chemical to a rat for 20 days, and after that, to check which P450s are induced, what you simply went into the liver of this rat and took out a sample of hepatocytes, and from that you got the mRNA. Okay. The third is pathology samples, especially when you're trying to look at effect of disease on RNA levels or expression levels, or biopsy samples, which dissection samples. So any of these samples which contain RNA can be used to do what is called as sample preparation, and typically this is what is done. Okay, if I'm doing just detection of a given sequence, okay, then what I will do is from that sample, since I want a very specific target DNA, I'll take the DNA and cut it into small pieces with what I'll call as restriction enzymes. And the DNA fragments that I get, I just label them. I label the fragments so that I can track each and every piece that I've made after I cut with restriction enzyme. The hope is some of these pieces will have the gene of interest to me, which will be complementary to the probe that I put on the DNA microarray. If I'm interested in looking at expression of a given gene, then I'm interested in the mRNA, which is present in the target cell. But because mRNA is generally unstable, and because the amount of mRNA in a sample may be very low to very high, if it is very low, it is very difficult to detect it. Okay, so what is typically done is you do what is called as RT-PCR, or what is called as reverse transcriptase PCR, where you convert the mRNA to a more stable DNA molecule, which is called as complementary DNA. And then what you do is use this DNA as what is called as a target to find out how it is changing, and when it is changing, it will reflect the mRNA which you have obtained from the cell. But before I use that copy DNA or copy RNA to do what is called my testing, I again will label it. Okay, so that I'm sure that I've tagged the RNA under question. Okay. So now if I do PCR, what will happen? Through this copy DNA, I'll get more samples of RNA, what is called as copy RNA. Sometimes what I might do is just do the RT-PCR many times and get only the copy DNA. Okay. Either which way, I'm indirectly analyzing for this mRNA. Okay. Now, how do I label? Typically, I can do labeling using radioactive groups or what are called as fluorescent moieties, so that I can track these pieces or fragments as I do my experiment. So the next step, as I say, is after labeling, you do what is called as hybridization. So you add this labeled fragments onto the DNA chip that you have already made by either pipette technology or Affymetrix or Nimblegen technology, and then allow the target and the probe to bind. Okay, I give it time, make sure the conditions are stringent so that you don't get too much non-specific binding. And after binding has occurred, you wash to remove all the unbound target molecules. Okay, and then you detect and quantitate where exactly binding has occurred. So that is referred to as hybridization and detection and quantitation of hybridization. Okay. So to give you an idea, let us say this is a gene chip and there is a probe which is present on the gene chip at a given location. Here is my copy RNA or copy DNA which I have labeled with biotin. Okay, so the label is shown here. So if this sequence is complementary to this sequence on the chip, then this target will bind to this probe and I'll get this double stranded structure. And because these double stranded structures are fairly stable, they will not get washed away. And now this orange or yellow piece has got little labels, right? And that label will then allow me to detect this hybridization, because now this label will be localized in this region. And what I now do is make sure I detect that label in some manner. In this case, what is called as a biotin label is detected using streptavidin, okay, or and phycoerythrin, which gives color. Now, you can also do it by other ways like autoradiography or fluorescence using laser activation, or you can also do mass spectrometry. Okay, but you have to remember one thing. Once you do this hybridization and detection, you have to remember the spot sizes are very, very small. Okay, so many of these probe cells can be as small as 25 microns. Remember I told you microarrays have radius which is less than 200, but this radius could also be as small as 25 microns. So if I have one small area where hybridization has occurred and I need to detect the amount of radioactivity there or fluorescence there, then I need to have a detector also which is commensurate with that size of the probe cell. Okay, and now remember one thing. Typically, the detector will be 1/8 of this size. Okay, and the reason for this is very simple. Okay, now imagine, so this is a screen here, and these probe cells are present. Let us say I'm going to put the dots here. Okay, one is here, then another one is here. The third one is right next to it. The fourth one is right next to it. The fifth one is right next to it, and I go along this, and there are thousands and thousands of such little dots. Okay, and I want to detect what is happening at each and every dot. If I keep a detector which is as big as the circle I am drawing, then what will happen when the detector goes to this region? It is going to detect everything that is happening in all these dots here, and I will never be able to find out what is exactly happening at this dot. So my detector essentially has to be smaller than the drop. Only then I'll be able to look at one drop, and then as I go from that drop to the next drop, my detector will actually see no signal for a little while, and then it'll start again seeing signal when the next dot is coming. Okay, so it's like, imagine for a moment that if I want to find out in a class how many people are bald, okay, and I'm going to put a little a camera which is going to go from the surface of the on the surface of the heads of every person. If I keep the camera about 4 feet in diameter, what will happen is it is going to catch about 30 heads at one time, and I will miss if somebody is bald in that, because as I move, I'll get many people who have got a lot of hair and maybe one or two who are bald. But imagine I do the other way. I make a detector which is smaller than the size of the head. Then what will happen as the detector is moving? You'll see hair, 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 hair. Then you'll see gap. The gap is between two heads. Okay, but since the detector is smaller than the heads, it'll actually see the gap. Then it'll go to the next head. Then you'll see little tuckle, tuckle, tuckle for a little while. Okay, because this guy's head has got no hair anywhere. Okay, then after it passes there, again it'll reach a point where there is nothing. Okay, you'll see no signal. Then again hair will appear. Okay. So you have to imagine that the detector cannot be bigger than the size of the probe cell, so that it can detect where all these binding events have occurred. Okay, and because of that, you can also get sometimes what are called edge artifacts or a signal to noise ratio. So these are all technical issues. Okay, but those are all not our headache. Okay, the technical fellows will take care of that. The important thing is to remember that things are not as simple as you think they are. Okay, when you miniaturize things. Okay, so again, as I said, this is a broad, sequence of what is going to happen. If you go very specifically, here is an example of a DNA microarray chip, which is 1.3 centimeters by 1.3 centimeters. Okay, that is what it's saying, 13 millimeter by 13 millimeter. But the way the spots are set up, there are 400,000 spots which have been etched on this plate. Okay, and if I were to go to any one location and magnify, I might see all of these little fields here, and each is shown to be different because it has got a different color. For example, this has got a blue color, that means it has got a certain sequence of nucleotides. In here it is red, it has got a certain sequence of nucleotides. Here there is a green, and so on and so forth. Okay. Now imagine now in my sample are these labeled complementary RNA or DNA which are going to bind to this. Okay, and for a moment imagine this pink is going to bind to the red, and this light green is going to bind to the green. Okay. Now see what happens when I throw this RNA on this microchip. What these pieces will do is go exactly to this location where they will spot these sequences, and when they spot these sequences you'll find that they'll complementary bind to that location where the red is. Okay, now remember here I showed red is going only one. Actually speaking, if I go very close, the red is going to have millions of molecules there. Okay, and this will go and bind there. The light greens will go and bind here. No binding will occur in this blue region because there is nothing complementary to that. And now what will happen? Because I've got a little label here, which is tagging all these things, I'll find that the signal in this region will be equivalent to these two labels. Here the signal will be about 2.5 times that. Okay, because there are 5 such labels, and the signal here will be nothing, 0, because there is no label. And that will be read out in the detector as being 0 or little bit or little more than that. Okay. And that is the advantage of labeling and using a photograph. Okay, so see, if these things are so close to each other, my detector has to be very small and has to first go to this region and then later on goes to this region and then it will go to this region. And when it does so, it will then see a no signal and then some signal and then even more signal. Okay, so those instrumentation parameters are very crucial. Okay, and in reality, what is happening at any very specific location is this is a base on the probe, this is a base on the target. There's exact matching here, so you get hybridization, and these labels help you detect that formation of this double-stranded structure. Okay, again shows here in the same fashion. Okay. Now, what typically people will do is that they do something very intelligent. When I'm looking for differences in gene expression, I invariably want to compare it with what is called as a control. Okay, just to give an example, if I want to do induction of P450, okay, so I might have a group of rats which are not treated with anything, and they're treated only with vehicle, and then another group of rats where I'm actually treating with the compound I am thinking that it may be inducer of P450, and I do that induction for 5 or 6 days or maybe 10 days. Okay, and then after that I sacrifice animals from both groups. Okay, now what I do is very interesting. Let us say I'm looking at P450, then what I would typically do is from the control animal I would take hepatocytes, break open the hepatocytes, and try and get at the mRNA. Okay, and take that mRNA and make what is called as copy DNA or copy RNA and amplify it by PCR. At the same time, I'll also take a liver from the test animal, which has been treated with the chemical in question. There also I isolate the hepatocyte, get at the mRNA, amplify it. Now see what I do very interestingly. When I label, what I do is I label the control mRNA with a red tag, and I label the sample mRNA with a green tag. Okay, now see what I do. On this probe here, on this DNA chip as it is called, are a lot of probes whose sequence is complementary to the genetic sequence of all your P450s which are present in humans. Okay, for example, we have 57 P450s which are known to be present belonging to different families. Since I know the gene sequence of all 57 of them, I can make probes which are such that they will detect only one of the P450s and not react with the mRNA which is obtained from any other gene of any other P450. Okay. Now, because of that specificity that I have, I might put all the probes here for the different 57 P450s, say in this location. Okay. Now, remember, there are so many fields here that I can put many different probes. But since in this very small experiment, I'm only interested in P450, those 57 probes will end up only in this very region. I don't even have to use the whole chip. Okay. But see what I do. The RNA which I got from my control, I label with a tag which gives red fluorescence, let us say. And the mRNA which I've obtained from my sample, I tag it with a fluorescent signal which gives green fluorescence. Okay, just for an argument. Now what I do is I mix both the RNAs and allow them to interact with the sequences which are present on this chip. Okay. So what I might find is that let us say here this is the sequence for CYP1A2. Okay. Uh, so all the mRNA which is present in this sample and this sample, which is complementary to the sequence of 1A2, will come and hybridize here. Okay. Now, the interesting part is if there is no induction at all of 1A2, then the amount of mRNA that I get here and the amount of mRNA that I get here will be the same. So when I mix both of these and I throw it onto the probe, the signal that I will get at the location of 1A2 will be somewhat like this, two reds and two greens, telling me there is no change in the expression pattern. Okay. But I might also find that at some locations there are more greens than red. That means what? That mRNA is present in a larger quantity here than here, and that is reflected by more green and less red, which means that DNA or that P450 has got induced. Likewise, I might see the opposite trend also. There is more red and less green. That means something has happened and this chemical has caused repression of a given P450. So I can actually detect no change, increase or induction, and repression by simply tagging it differently and mixing, and I do this analysis as a relative measure. Okay. So, and the reason we do it is very difficult to quantitate signals, but it is very much easier to look at relative changes. Okay, and in biology, most of the time we are interested in relative changes, anyways. Okay, so you want to say whether there is two-fold induction or four-fold induction, or let us say three-fold repression. Essentially, what we do is look at the relative amount of red to green signal, and that will tell me whether there is induction or repression. Okay. So usually this is the way it is set up. Rather than numbers, we look at color. Okay. So if it is mostly green, that means it's indicating of repression. If it is mostly red, it is indicating of induction. And then it is equal amount of green and red, which will actually end up as kind of black. Then you say, okay, there is no change in the expression. Okay, and this is how you get a feel for whether a given DNA is being expressed as mRNA or not. Okay, so here's a nice example. These are all RNA samples from different cancer tissues. Okay, obtained from different people. Okay, and I'm trying to see whether the same cancer. Okay, in this case, cervical biopsy, as obtained from different patients having cervical cancer, all have got the same level of gene expression, or they have got different levels of gene expression. And if so, does that correlate in some way with the propensity for their cure when treated with certain compounds? Okay. I do the very similar experiment. I will take what I call a normal uterine samples. Relative to that, I'll take tumor. I'll again extract the RNA, label differently, and every time look for the relative amounts of green to red signal. Okay, and I might find that my DNA array, which is looking for all the different genes which are associated with, say, uterine cancer, might look somewhat like this. Okay, at some places it is green. Okay, that tells me, for example, it is, say, repression. At other places it's red. That is telling me it is induction. At some places it's red and green and red mixed together to give kind of what is called a little bit of yellow. Okay, that tells me both are present. Black means there is no signal at all in that region, so that mRNA is absent only. Okay? So we do that, and then basically in the end, for all the different genes whose sequences you have put on the array, you get to know what is the relative rate of expression. If this number is red, that means there is induction, in the sense the tumor has more of that gene mRNA than the reference or the wild type sequence. Okay, and where it is the opposite, it is green. That tells you that there is what is called as, let us see, green. That means it's the opposite. There is repression. Okay. Now, typically these are done by computers. So since you know exactly where is what, in the end you might get something like this. That when you do an experiment, after 2 hours you see this profile, after 4 hours you see this profile for the different genes. So it tells you now how, let us say, it is getting induced and then coming down, or it is staying induced, or it is getting repressed and then coming back. Okay, and so on and so forth. And then we use our algorithms to do what is called as clustering, so that you can kind of get all the similar looking genes whose expression is going up, all located, and we can do what is called as very typically what is called as principal component analysis, or analysis of variance, how one expression increases with another, and so on and so forth. Okay? Now, what you have to remember is when you do this microarray, and if you remember, if you have a chip which is very, very small, anything from 1.2 to 1.2 centimeter, and you have got 500,000 probes. If I do one experiment in duplicate, at each of this probe, I'll get one data point. So my number of data points will be 1 million. And if I do 5 experiments in duplicate, I'll get 5 million data points. Okay? Now remember, when I'm generating so much data, I have a lot of issues. Am I getting the data correctly? Is it accurate? Can I make sense of this data? I can't just look at it and figure out what is what, because these are little spots of red and green and blue. So I need to have systems which will allow me to handle this much data, and also I have to have good algorithms which will allow me to look for differences and even correlation of expression. So this microarray technology comes with a big headache, that you have to have equally good, what do you call the data analysis software, and data storage capacity, and data handling capacity, so that you can use this microarray technology efficiently. Okay. But the good thing is, it is there in the market. It can allow you to do so many things. It can allow you to do tissue typing, polymorphism, disease detection. You can look at expression pattern after drug treatment on the host. You can look at expression pattern after drug treatment on the target. You can do expression pattern during multiple therapy, associated with disease, associated with toxicity, pattern of drugs which have a specific toxicity. You can also use it in agriculture, in food industry, you name it. Sky is the limit. Okay. But the problem is, you need to know something about the genome so that you can design the probe. So in that regard, it's what is called a closed system. So unless you have information of the gene, you cannot make a probe. Okay. The second thing is, it is expensive. If you buy what are called as off-the-shelf chips, okay, where they have put certain probes based on their liking. Okay, it's like getting cable with a certain package, which have got certain news channels and certain entertainment channels, certain sports channels. That tends to be a little cheaper. But if you want a custom-made chip, that means you do a la carte. Okay, I want this channel, I want exactly this channel. Then it starts to get costly, and it ends up being about 5 times more costly. And all these chips you cannot reuse, single use, and the detectors and the readers are also very costly. Okay, $500,000. Then this is about 5 to 10 years ago. It is probably cheaper now. Uh, and then there's always this question of how accurate these are. Okay, but apparently they give very good quality data, at least in a relative sense, and the cost is also getting less and less. Okay, and now this is the beauty. 100,000 bits of human genetic code is there on a single chip. All E. coli genes, all yeast genes, they are put on a single chip. So I can do an experiment on E. coli and look at every gene, whether it is expressed, increase, or expression is increase or decrease. Okay, uh, all known 6,005 human genes. Okay, this is not a right number as of the moment, because the number of human genes is close to 20,000 now. Okay, uh, but there is also chip available with 42,000 predicted genes. Okay, based on your computational analysis. Many polymorphisms you can detect. You can detect a lot of mouse genes, okay, rat genes. And Affymetrix proposes to have 1.6 million probes per chip. Okay, at the moment the number is around 500,000. Okay, so they want to make it about 3 times that. Okay, so 1,500,000. Okay, on one chip. So you can imagine how many different genes change in expression levels you can look and find at one shot. Okay. Now, again, here is some more things. All of these are all targets for to put on the gene chip. Okay. So at one point, they are thinking that you could also have what is called as a chip with, in a sense, the DNA of all the pathogens. Okay. So tomorrow, when you suffer from some disease, no, the doctor might take a sample of your blood, or say urine, or a biopsy, or a exudate or something, and throw it on the chip, and the chip will tell you you are infected with what, because it will detect the DNA of every known pathogen, or RNA of every known pathogen. It's called lab on a chip kind of thing. So now you don't have to tell the doctor test for TB, test for malaria. You just say test, it will test for everything. Okay, so so at one point these days will come where when the doctor says go get a test done, they will not specify what test. They'll just say do a chip test and let me know what all has happened to this person by looking at the sample. Okay, so here are the companies which are involved in this technology. Some very classic players. Okay, Affymetrix is the leader in this chip fabricator, but all of the very famous companies like HP and all, they have all got into this at some point. Sometimes you don't recognize them because their parent name might not appear, because these are all what I call as subdivisions of the main company. Okay, for example, here is Amersham Pharmacia. Okay, so it is part of Upjohn, and this is New England Nuclear, and so on and so forth. Okay, uh, but then all the big players who are in this sequencing and all, they are also in this microarray market. Okay, so the idea is that if we do these things correctly, then we might know how a cell is working. So this is typically how different things interact inside the cell. Okay, and even worse is when EGF binds to EGF receptor, epidermal growth factor receptor, this is all that can happen inside the cell. And one can look at the expression of all of these proteins and how they change in one shot using this technology. Okay, so basically at one point there will be integration of all of this, uh, changes in mRNA, changes in protein, transcriptomics, metabolomics, and they'll put together all this, and you'll then understand your system much better as part of this area of study which is called as systems biology. Okay, so here are some references from where you can get these things. And when I prepared this, I had a lot of help from a lot of people. Uh, Bhuvan, my wife, Lata and Advait, who are students from BCP. Some sites I got from Dr. Chandan Kumar from ACTREC. Okay, and of course BCP. So there we go. So if you have any questions, you can shoot. Okay, so I'm going to stop sharing and go to my let us say escape here. Go back to my screen, and there we go. So any questions you have, any queries you have, go for it. Hopefully, I'll be able to answer a few of them. Okay, yeah, Introduction to Proteogenomics from Sanjeev Srivastava. Yeah, he's there at IIT Mumbai, and he does a lot of this. Okay, and he's into a lot of this development of analytical technologies. Okay, so after you do the experiment, no, how to do correlation, how to do cluster analysis, how to do what is called as principal component analysis. He's an expert in all that. Okay, so his area is actually making sense of data. Okay, when you use this array technology. Anyone else? Any questions? Anyone?
Sir, what I feel a variant of that technology is still available. Like we have many pregnancy detection kits, like you spot a drop on it and then it can detect like what is there or not there. A type of
Yeah,
it, I can say.
yeah. It's actually a type of microarray technology, but it is very specific for a single entity. Okay, uh, where you have attached maybe antibody on a surface, and you're looking for a very specific antigen to come and bind to it. What we have learned as ELISA or RIA or what is called as that type of technology. Yeah, in a sense, that is kind of what is happening. You have a very specific interaction, but what you have done here is you have set up a system where you can look at thousands of different interactions at the same time. Okay, and that is the power of microarray technology, where you can do 1,000 different tests in one shot. Any other question? Anyone? Sandhya looks silent today. क्या हो गया संध्या?
No, was wondering about the probe, but in the last slide or second last slide, you mentioned that you need to know the sequence to actually design a probe. So it wouldn't be useful if you have some unknown
Yeah, yeah.
target, right?
Yeah, you just can't. Yeah, you can't just put any probes at random. What
Mm-hmm.
that will do is it'll detect something which binds to it, but you'll get no information from it, except
Mm-hmm.
that there's a sequence complementary to this probe, but you're interested in how it relates to a certain gene. So
Correct.
you do need information about the genome. Okay, only
Okay.
that.
And what is this light source that they use in the lithographic method, which
Those are all
activates?
very specific lasers. Okay,
Okay. Mm.
so which have a certain energy content,
Uh-huh.
and at that wavelength, you get this deprotection reaction. Okay,
Okay.
so you use very specific wavelength of light. Typically, lasers are used so that they don't scatter too much because you want it to be very localized.
Correct.
Okay, so yeah, so very specific wavelength lasers.
Okay.
Anything else? So hopefully you learned something new today. Uh, how many of you had an inkling of what this was before we started, and how do you feel now? Yeah, okay.
Sir, what I feel like enzyme induction, if we go to measure in our lab, it is much cheaper. Na? We just take a substrate and measure its conversion. If it's faster, we call it induction. If it is slower, then we call it as repression. So
Correct.
currently, our technology is much cheaper, I believe.
And how much time does it take for our technology to work, Sahil? It is typically M-Form project, right?
Yes.
I give to 6 rat, then I isolate the liver, and then I do this catalytic assay. Okay,
Sir,
if
you
I
missed the microsome preparation.
micro preparation, and then you do this assay and you look for catalysis. Hopefully, your HPLC will work. Uh, what Sandhya did in 6 months can be done in 10 days. Okay, and the reason 10 days is because 9 days you have to dose. Okay, the actual getting the hepatocyte and getting the RNA and doing the experiment is a day and a half. Okay, so where do we look at cost? Okay, how do we analyze cost? The cost of your 6 months versus the cost of you know something which just has a tag of 500,000. Imagine every day an M.Pharm student will complete his M.Pharm. Or actually, if I do it correctly, no, if I make the chip correctly, I can have about 500 to 600 M.Pharms ka project being done in one day. Okay, because I will simply pick samples from different sources such that they have to interact with different probes, and all those probes are there on the same chip. Okay, so one corner ka data will be of student A, the other corner ka data will be of student B. Okay, so see, cost wise, what we are talking is a very odd way of looking at cost. Okay, sometimes what we feel is I don't want to spend $500,000, but I'm willing to spend a year and a half to get that data, which this fellow will give in a day and a half. So which means year and a half of my time is less than $500,000, and that might not be something which you'll be able to calculate also. Okay, so this way of looking at things, is see at the M.Pharm level and all. We tell you to do this because we want you to learn some of the things which occur at the background when you start using technology. Okay, and so that you realize what technology can do for you, and a little hands-on is required before you can start using the technology, so that the technology will not fool you later on. But but it's not that we are it is cheaper. It is not really like that. Okay, you can ask Sandhya only. She has seen both sides of it. She has seen the lab, she has seen Nicholas, and she has seen BMS. So when she was doing her M.Pharm, okay, she and Madhav used to get access to the best instrumentation, and they never used to wash their pasture pipettes and test tubes. Just throw them after they are done. At the same time, they were doing their work. Who was there in the lab? Sandhya, along
Where at? College or?
Yeah, when in your M.Pharm batch, you and
Mm-hmm.
Madhav
Uh-huh.
were the Nicholas, but I
Correct.
think Sham was at the same
Sham,
time.
Sham was it? Yeah.
हाँ, तो शाम रोज टेस्ट ट्यूब धोता था, ये लोग टेस्ट ट्यूब करते
Perfect.
so so
The
so so
filter paper ka, we would actually when we came back
hmm,
for mobile face filtration, we would use a filter paper and then throw it. And then I remember Rohit getting all wild and asking, "What you come from America or something that you're using a fresh filter paper every time? You just
हाँ
wash it and reuse it. You don't throw it like that."
हाँ तो इधर कैसा था, मोबाइल मोबाइल फेस का जो फिल्टर पेपर था ना, और मोबाइल फेस का दैट मेम्ब्रेन फिल्टर, इट वाज नॉट फॉर ओनली वन 200 ml to 2 liters ऑफ मोबाइल फेस, उधर वो अपना आजकल का वो टाटा का प्योरिट आता है ना, वैसा था, 600 लीटर मोबाइल फेस जाने के बाद फेंकेंगे हम, वैसा नहीं तो नहीं फेंकेंगे उसको. So it came with a volume of mobile phase which could be poured through that filter before you discard it.
And
So,
reversely, when I see a vial of microsomes, especially rat or mouse liver microsomes, I remember my homogenization days as to people are just ordering it and they would sit and chop, make those buffers and actually homogenize. And
get delivered, yeah, and
yeah,
spend the whole afternoon, and
yeah.
after that keep it in the freezer and guard it with your life. ऐसा बोलते हैं. ठीक है. Okay. Any other questions? If not, uh, enjoy the week. Hopefully, we'll meet again next time with some other new topic. So I'm going to try and see whether I can come up with topics which are all new to you. Okay, and in between, I'll also give DMPK. Okay, but since we do DMPK all the time, I thought I will change ship a little bit for a little while. Okay, so shall we? Bye bye, Karu. Any other questions? Anyone before we close? Okay, nice to have you all. Nice to see you all. Should we do like last time? 1 minute के लिए we'll turn our camera on and say hi to each other. So here I go. Here I am, hopefully visible to you guys. Let us see. Hi, hi everyone. Hi Girish. Hi Deepali.